Okay, so thank you, Dr. Mona Lisa, for that introduction, and a very good evening to everyone, the chairperson, and all my other colleagues who are there with us for this talk today. So, actually, I thank the organizers again for thinking of this topic. Fact remains that HLA, as a topic itself, is quite challenging, and it's quite complex to understand. So, I have tried my best to make it just simple, and I would mainly focus on the clinical applications of HLA based testing. So this is the outline. We will look at a little bit of introduction followed by history, and then a little bit of the nomenclature, the various structures, proteins, etc., the properties of HLA molecules, the functions of HLA, and then we will look in details about the technology that is available. And we will discuss the pros and cons of each technology that is prominently used in the laboratory today. And then before I close, I'll talk a little bit about organ donation and our role in acceptance of the donated organ. So when we talk about HLA, it begins with the immune system, which I think all of us read about during our pathology training, etc. So I won't go much into the details of that, but we all are aware of the various organs that are involved in our immune system. The bone marrow being one of the major organs, the lymph nodes, the spleen, and various other lymphatics, etc. So when we talk about HLA, it involves the immune response, understanding the immune response, and anything that can cause an immune response is known as a antigen. Right. So here we are going to focus mainly on understanding the major histocompatibility antigens. So in fact, a better way of understanding the HLA or the human leukocyte antigen, or the better terminology today, is MHC, major histocompatibility antigens. And these major histocompatibility antigens are integral to the normal functioning of the immune system, right? And HLA is, of course, as I said, a part of the major histocompatibility antigen. So let us now try and understand the concept of histocompatibility, basically. So whenever we talk about compatibility, we know that it is something related to matching or compatibility or agreement between a donor and a recipient, right? So in the scenario of transplantation, may it be a bone marrow transplant or a solid organ transplant, what we refer to is the ability of the donor's organ or tissue to be accepted by a recipient. So that is histocompatibility, right? And as I said, the group of genes that are present in all animals which are responsible for this histocompatibility or play a major role in the histocompatibility would be known as the major histocompatibility or the MHC complex. Okay, so let us look at the discovery and a little bit of the biological scientific significance of these. So we can't progress further without understanding the contributions by these great scientists. Way back in 1936, we had Peter Gorer, who did a lot of work in understanding the MHC complex and the basic concepts of the HLA. Then we have, Dr. Dawsey, then John von Rood, Walter Bodmer, a lot of them, I could only name a few of them over here. Terasaki, you must have heard about Terasaki plates, and we will look at them a little later. So these are the scientists who have tremendously contributed to the field of HLA. And that is how today we are at least able to define and understand a part of it. I would say that I'll again come to that later, why I'm saying that we are just understanding a part of this concept of HLA. So let us try and understand what exactly is HLA. It is a set of molecules which are essentially glycoproteins, which are expressed on the surface of almost all the cells that are responsible for being recognized or which are involved with antigen presentation. Okay, so the HLA molecules basically will control the immune response through this recognition pattern or function of recognition. So by this function, they will either recognize the particular protein or tissue as self, or they will not recognize it and treat it as non-self. So basically this is just acceptance or rejection of a graft. This is the basis. So these cell surface proteins, they're responsible for the regulation of our immune system and its response in humans, like all other animals. So where is this located? Precisely, it is located on chromosome 6. Okay. What we also need to understand is that these HLA genes are highly polymorphic. What do we mean by that? They are highly polymorphic is that they will have many different alleles, and therefore they can fine tune the adaptive immune system. Okay, so they will not be in one fixed form. They may change their forms. They will have different alleles, and you will see different alleles in different situations in the particular individual. Okay, let us look at the classification of these HLA. So this is just very bare basic, which I thought I should include over here. So basically, when you look at the MHC, you have the class 1, the class 2, and class 3. Okay, for all practical purposes, we consider the class 1 and the class 2 major histocompatibility complex, or the HLA class 1 and class 2. The HLA class 1 will include HLA A, B, and C as the most dominant or the major components, whereas there are certain minor components like the HLA E, F, and G also. But in the practical testing, we look at A, B, and C HLA when we talk about class 1, whereas the class 2 includes majorly the HLA DR, DQ, and DP, whereas on the minor side, we have the DM and DO also that are present. Now, what we need to understand is the MHC class 1 are present on all nucleated cells. right? So all each and every nucleated cell will have a expression of class 1 antigens, and the function of these MHC class 1 or HLA class 1 will be identification and destruction of abnormal or infected cells by cytotoxic T cell production. Okay, so we have to understand the mechanism here. This is cytotoxic T cell related activity, right? So that is by presenting those infected cells to the CD8 positive cells. Whereas when you talk about HLA class 2, they are typically present on the B lymphocytes, on the macrophages, on the dendritic cells, activated T cells, and the endothelial cells. Okay, so class 1 on all nucleated cells, class 2 on B lymphocytes, macrophages, dendritic cells, activated T cells, and endothelial cells. And the function is identification of foreign antigens and presenting it to the helper T cells. So in class 1, it was cytotoxic T cells and CD8 positivity. And here it is helper T cells and CD4 positive cells. As far as class 3 is concerned, that includes the complements C2, C4, various factor B, right? And 21 hydroxylase, TNFs, etc. So these are basically plasma proteins, and they provide defense against the extracellular pathogens by generating an inflammatory response and immune response. So this in a nutshell will give us an understanding of the HLA, the classification of the HLA system here, right? So now let us go into the structural aspect of it basically. So if you look at the class 1 molecules, these will contain, so you are looking at the class 1 molecules over here, so there will be glycosylated heavy chain that are coded by the HLA class 1 gene, right? So there is a alpha chain that is anchored to the cell membrane. So you can see the plasma membrane, the cell membrane over here, and you have the alpha chain. There is alpha 1 and alpha 2 domains which form the antigen binding cleft. So this is the most important part where there is a peptide binding groove, right, for class 1. And as we saw, these represent the this will be responsible for the binding to the T cells, right, binding of the antigens to the T cells. Whereas when you look at class 2, the structure is different over here. So these are heterodimers of two non-covalently attached glycosylated polypeptide chains. So you have a alpha chain and a beta chain over here, and alpha 1 and beta 1 domain, they form the antigen binding cleft over here, right? And we have also seen that class 2 will be interacting through the CD4 positive cell mechanism, right? Now let us look at the nomenclature. I'm sure if you have got a chance to look at the HLA typing report, you will see that the typing report consists of a lot of alphabets and numbers or figures. Okay, so let us try and understand what exactly is the meaning of each of these components, right? So the first HLA is the prefix, as we know, the human leukocyte antigen, and then that will be followed by a hyphen that is used to separate the gene name from the HLA prefix. Then you have either, in this case, it is HLA-A. So you're talking about the A gene over here. Then there is a separator over here. So this asterisk is known as the separator. Then you have the next figure, that is here you have O2. So this is HLA-A, O2. Now this is the allele group that is represented over here. Again, then there is a field separator after that. And next you will have 101. So that is the field 2, that is the specific HLA protein. So here you were at the level of the gene first, that is HLA-A. Then next you are defining the allele, and now you are defining the protein, which is again separated by a field separator. And then you have a synonymous DNA substitution within the coding region. So O1 will represent the substitution within the coding region. Again, there is a field separator. And then you have O2, which is used to show the differences in the non-coding region. And then finally, there is a suffix that is used to denote changes in the expression. So you don't go much into the complexity of this, but what we need to know over here is that when we talk about HLA typing in the practical day-to-day laboratory practice, we look at the resolution, level of resolution. So when you talk about HLA-A, O2, that will be the low resolution typing, okay, because you are looking only up to the allele group. When you go further, you have the intermediate resolution, the high resolution, and things like that. And today, so we'll later on see when we talk about technologies, how we have high resolution HLA typing, and for various clinical applications, what resolution do you need to test up to, right? So this is the baseline of understanding your HLA nomenclature. Then a little bit about inheritance. So when we look at the inheritance pattern, we know that this follows the Mendelian inheritance over here, and the expression of these MHC alleles is codominant. So what do I mean by codominant? Is that the protein products of both the alleles at a locus are expressed equally in the cell, and both gene products can present antigens to the T cells. So both get an equal chance of expressing their particular themselves, and also for presenting the antigens to the T cells, right? So the particular combination of MHC alleles found on a single chromosome is known as a MHC haplotype. Many times we talk about a haplotype match. So this is what is the haplotype. Okay. Now, again, to understand the inheritance, if you assume that there is no recombination, then 4 different haplotypes are possible in the offspring. So here you can see that these are the parents. So one of them is having A and B, and the other is expressing C and D. Then what are the possibilities would in the progeny that are given over here. Or you can look at this table where in details you have the ABC, DR, DQ and DP expression, okay, and the various haplotypes that are possible over here. So just to keep it simple, the chance that two siblings will be genetically HLA identical is 25%, right? And there's also a formula. So the chance that any one patient with n number of siblings will have at least one HLA identical sibling is 1 minus 3 divided by 4 into n. Okay, so this is just a formula. Don't really have to remember it. Now, when we talk about inheritance, we should know that the probability will never be 100% for finding an HLA identical sibling. Okay, so if you have two siblings, then it provides around 44% chance. If you have three siblings, it provides about 58% chance that one sibling will be HLA identical to the other, right? So each time a new sibling is tested, then the new sibling has about 25% chance of being a match. Okay, no matter how many siblings have been previously tested. Okay, so we have to just remember these probability rules basically when we look at the inheritance. Now let us come to a understanding of the diversity, right? As I said in the beginning that there's a lots that we don't know in this particular subject. Okay, that is because of this gene diversity. So as we said, MHC is polygenic. That means it contains several different class 1 and class 2 genes. Okay, so that every individual will possess a set of MHC molecules with different ranges of peptide binding specificities, and therefore it becomes polygenic. Also, the MHC is hyperpolymorphic. That means there are multiple variants or alleles of each gene within the population as a whole, right? And the MHC genes, in fact, are the most polymorphic genes. So you can see the schematic representation over here where we have described the polymorphic nature of the MHC gene. Okay, so again, just for you to understand, so few would be highly polymorphic. So you can see here the pink ones, HLA A, B, C, very, very polymorphic. So very important also clinically. Okay, so that is what we have to understand. Whereas, not highly polymorphic, but yes, definitely polymorphic are those ones represented in the green color. So DP and DQ in the class 2 are also, moderately polymorphic. The blue ones are oligomorphic and the yellow ones are monomorphic. So based on that, you can understand the clinical significance or the level of activity, of the various class 1 and class 2 HLA isotypes. Now, this is just for us to understand the unknown factor in the field of HLA study, MHC, and its testing. Okay, so here you can see that the total number of new HLA antigens that were defined every year has kept on increasing year by year, and it is on a rise because now we have a good understanding of the molecular platforms. Various molecular platforms are available, and a lot of studies are going on in this field of HLA or histocompatibility and immunogenetics. Right. This graph also shows that the international nomenclature mechanism of various antigens and alleles. You can see over the period of 1968 to 2008. So in the 20th century, it has shown a sharp rise in our understanding of the various class 1 and class 2 alleles. So every year something new comes up and there is a update for us, and that we need to keep ourselves abreast with whatever new has been reported in the field every year. Right. Now let us come to the actual practical aspect, that is the various techniques or the methodologies that are used. Right. So here basically this is just the broad classification. So we began with serology. Like we have immunohematology and serology. So serotyping was there, where non-sequence based typing methods where antibodies specific to the HLA proteins were used. So you have different HLA proteins that were known at that time, and you detect the antibodies to those particular specific HLA proteins. That is the serology method of testing. Then we had the SSOP, that is sequence specific oligonucleotide probe. So this involved PCR product that is tested against a series of known probe sequences. Then came the SSP, that is sequence-specific primers, and this technique allows the amplification of specific fragments of the DNA to produce millions of fragment copies, which can then be further analyzed by your gel electrophoresis. Then we had the Sanger sequencing or sequence-based typing, SBT, where there is a technique for DNA sequencing based upon the selective incorporation of chain terminating various nucleotides, dideoxynucleotides by DNA polymerase during in vitro DNA replication. And the last and the most focused today is the NGS or the next generation sequencing, where what you do is perform long range PCR to amplify the HLA genes. Then you fragment those HLA genes and you perform deep sequencing of individual fragments that have been made of the particular HLA gene, right? So there are various equipments that are used for this HLA typing, and the most common ones I will include in the next slide. But before that, what we need to understand is the basis of transplantation. So now we are closing towards our actual practical application of the knowledge of HLA and MHC. So class 1 and class 2 matching is very, very important in organ transplantation. We have seen the most common solid organ transplant that is done is kidney, followed by liver and heart and pancreas and intestines, etc., and lungs. And of course, bone marrow transplantation also has been practiced since many years now. The mismatched HLA antigens may result in a very strong cell-mediated immune response, and these responses, they contribute to acute rejection in organ transplantation and GVHD or graft versus host disease in your bone marrow transplantation or the hematopoietic stem cell transplantation. So let us look at the various methodologies for antibody testing, HLA antibody testing. So the first and foremost, I'm sure you must have heard about this gold standard, still considered as the gold standard, CDC cross-match. Okay, so here what you do is you have the patient's serum and the donor cells, and if donor, you test them, and if the donor reactive alloantibodies are present in the patient's serum, then these antibodies will bind to the donor cells and then they will activate the complement cascade, leading to cytotoxicity. So what you look at the end point of this testing, CDC cross-match, is the cell lysis. So we'll come to that a little bit in details. Then there are tests that involve HLA antibody screening, where you can have a qualitative detection of the presence or absence of various HLA class 1 and class 2 antibodies. Then there is a terminology which you will very often come across, DSA, that is donor specific HLA antibodies. So there are tests where you can qualitatively detect the HLA antibodies against any specific donor antigen, right? Then SAB, that is single antigen bead testing. So this detects specific antibodies against class 1 and class 2 antigens. So if I have to give you an analogy, if you look back, to immunohematology principles, there is something known as antibody screening, which is just a general screening. So you get a yes, no result, whether a patient has got red cell antibodies or not, you do an antibody screening. So that is DSA, donor specific antibodies. But if you want to know what is the specificity, the antibody screening is positive. Now you want to know whether it is an antibody against any of the RH antigens or KEL antigens or KID or Duffy or MNS or whatever, then you do a 11 cell panel. So here similarly you have the single antigen bead and you will know exactly against which HLA antigen there is a antibody in your recipient or in your patient. The last is panel reactive antibodies, which represents the percentage of reactivity for a particular patient against a population. So this is just a kind of a percentage probability of finding a match in a given population. Okay, so let us go further and see what exactly these mean. Yes. So let us look at the CDC first, the complement dependent cytotoxicity. If I can say here that in India, even today, all our nephrology colleagues, they demand a CDC cross match in most of the sectors. Yes, there are examples of various cities or various centers who have tried to do away with the CDC cross match because, its positive predictive value is good, but the negative predictive value is very poor. So they have replaced CDC cross match either with a flow cross match or a Luminex based cross match, solid phase cross match, etc. But majority of the centers still require, and legally CDC is still required. So in CDC, what we do is you have the donor lymphocytes that are isolated and you have the patient or the recipient serum. You react them along with various other things, reagents, you fix them with formaldehyde, etc. And finally, there is addition of complement. So as I said, if there is antigen and antibody reaction and binding, that will activate the complement and that will lead to lysis of these cells, which you can actually see under an inverted phase contrast microscope, right? So this is just a pictorial view over here. So what you do is you score the reaction of lysis. So if the percentage cell death at the end of this CDC cross match, if you see that about 10% of the cells are dead, that is acceptable, and the interpretation of your cross match will be negative. If you find anywhere between 11 to 20% cell death, it is a doubtful negative test result and you may want to repeat the test. If the cell death is anywhere above 20% and up to 50%, you report it as a weak positive CDC. From 51 to 80%, it is a definitely positive CDC cross match, and 81 to 100, it is a strong positive CDC cross match, right? So you can see over here, this is how the dead cells will appear. Okay, a sheet of flat lying cells, whereas the live cells will be kind of translucent and they will be appearing rounded and not flat in their shape. So with training, you develop an eye to interpretation of the CDC cross match results. Believe me, it's sometimes for the beginners it's very dicey, and therefore many people do not rely only on the CDC cross match result, but you do another test as a supplementary test and combine the results, interpret both the results, a solid phase test and a CDC test, and you use that for your clinical decision making, right? So again, CDC cross-match was typically done with whole lymphocyte, but now there are various accreditation requirements that you at least, in order to increase the sensitivity, at least separate the T cells and the B cells, and then you do your cross-match separately for T cells and B cells. So the interpretation over here, if both your T and B cell cross-match is negative, that means you can relax, there is no donor-specific antibody, or the antibody may be there, but in very, very low titers, okay? If both T and B cell cross-match is positive, that means you definitely have a class 1 as well as a class 2, or a very strongly reacting donor-specific antibody of either class. If your T cell is negative and only the B cell cross-match is positive, then there are definite antibodies to class 2 or low level of donor-specific antibodies to class 1, sometimes non-specific antibodies, or maybe some monoclonal antibodies have been used like rituximab for the treatment. So history taking is very important in this case while interpreting the results, right? If your T cell cross-match is positive and B cell is negative, then definitely there is a possibility of any technical error, or the viability of the B cells may be questionable and you may want to repeat your test. Okay, so there are certain advantages. As I said, this is like a mini transplant. So you are kind of mixing the cells as well as the serum, and before actually putting in the organ, you are confirming whether there is any reaction or not. Okay, so as I said, this remains the gold standard test for solid organ transplants even today, and we continue to do so. But definitely there are limitations like labor intensiveness. It takes about 4 hours to complete the test, and at each step you have to be, there are various timelines involved. You have to be very, very specific in dispensing the reagents, etc. The cell separation is difficult, isn't it? Another important consideration is only complement fixing antibodies can be detected. If you don't have complement fixing antibodies, then they may be undetected in this case, right? And then there is a lot of interference with other substances like solvents, microbial products, toxic substances, detergents, etc. Right? So all these things you need to bear in mind while reporting your CDC test results, right? The next very commonly used platform is solid phase testing. And I'm sure you all must have heard about the Luminex technology or the XMAP technology in the solid phase assays over here. So basically here, what is done is these kits, they have various beads that are coated with the various class 1 and class 2 or a mixture of class 1 and class 2 various antigens depending upon what you want to detect. So whether it is a class 1 test for class 1 antigens or class 1 antibodies or class 2 antibodies, based on that you will have various panels that are available, and you have panels for single HLA antigen also, like I talked about the single antigen bead test, right? So these are all recombinant antigens that are coated, and then you add the patient's serum and you look for the reaction basically. So here what you need to understand is the limitations of these assays basically. So you have a something like our blood cross match, you have a Luminex cross match, which is like just a broad spectrum test where you prepare the donor cell lysate and you add the patient serum and you look for any donor specific antibody. Then you have the panel reactive antibody test or the HLA class 1 class 2 antibody screening that is done, and then finally as a confirmatory test you have the single antigen bead test, right? So as I said, the order of sensitivity we need to understand. So basically a DSA cross match or PRA screening would be a baseline test, whereas the confirmatory test would be the most sensitive, would be the single antigen bead, because that will give you exactly what specificity that antibody has, right? So there are certain precautions that we need to take or limitations that we need to understand over here. First and foremost, any contamination of the test material, inadequate incubation period. So you have to be very, very specific about your incubation temperature, the periods, etc. Over or under incubation can cause aberration in the test results. Inadequate washing. So adequate proper washing step is very, very important, right? Or exposure of the conjugate or various reagents to light. So when you remove those reagents, you have to see to it that there is no direct light or sunlight, etc. So you have to maintain a dark environment. The other technical limitation would be unable to detect antibodies to HLA-C or DQ and DP basically. So only if you have antibodies against the A, B and DR, etc., you are likely to detect them at the first go. But certain antibodies like DQ, DP may not be detected, or you may have false negative results because of the prozone effect. So you have to keep that in mind, and you can then have titers done, and you can dilute the serum and test again to rule out the prozone phenomenon. So what we need to understand over here is, why are we looking at these donor specific antibodies? We are looking at them because they mediate graft damage. Okay. So first here what happens is there is activation of the complement cascade, which will include C4D and C1q. Right. And when you look at the HLA antibodies, both HLA class 1 and class 2, those antibodies will react, and the non-HLA antigens on the cell surface also will be involved over here. Then you will have the neutrophils, macrophages, the NK cells, etc. that will be arriving on the scene, and there will be an antibody dependent cell mediated toxicity. So basically after the activation of the complement, all the class 1, class 2 antibodies as well as the other macrophages, neutrophils, etc. will lead to cell lysis. So this is the basic mechanism of any graft damage that occurs after a non-properly matched transplant. Okay, so here we can look at the various antibody detection methods in solid organ transplantation. As we talked about CDC, there is also flow cytometry cross-match. We'll talk about it a little later. ELISA technique is also used, which is generic or specific, and that can be used for various HLA antibody specificity testing. There is Luminex, which can be generic or also single antigen bead or specific testing that you have on the Luminex platform or the solid platform, right? So basically you have the serology, which includes the CDC, then you have ELISA testing, and you have the solid platform testing. Now, organ-wise, let us see what are the various methods for antibody screening and cross-matching, etc. So if you are dealing with kidney transplant, the most commonest, pre-transplant screening is definitely required. So pre-transplant, you look for the presence of any antibodies, etc. What you will do generally would be a solid phase test plus a CDC or a flow cytometric cross-match. Okay, so as I said, no single test is done, but you do at least a combination of CDC and flow or CDC and Luminex based cross-match, and take the both the results into consideration, compare it with your clinical setting, and then arrive at a clinical decision. Okay. So basically why this is done, it is to prevent any kind of acute antibody mediated rejection. In the heart transplant scenario also, you definitely have to do a pre-transplant screening and you use solid phase assays or CDC, but CDC is not mandatory as such, but definitely there are transplant surgeons who prefer to get a pre-workup done or a solid phase assay either just before the transplant or after the transplant also, so that they can modulate the immunomodulatory treatment or drugs. For lung transplant also the same principle is there. For liver transplant, basically there is not much of HLA based testing that is involved over here, right? So there are very few centers who really look at the HLA antibodies for a liver transplant also. For pancreas and islet cell transplant, yes, definitely that is indicated. And also for the intestinal transplants, definitely this is indicated. Okay, so now we do all these basic screening tests to understand the risk assessment. So here I would just want your attention to understand the risk assessment or the gradation. So suppose you have done a Luminex based antibody screening and the MFI, that is the mean fluorescence intensity. So that is the unit, the output, the test result will give you a MFI value, mean fluorescence intensity, and you have a cutoff. Generally the cutoff is anywhere between 500 to 1000 based on the standardization in your own laboratory. So if you are getting a MFI value of less than 1000 for both class 1 as well as class 2 and your T cell cross match or CDC is negative, then that means you are essentially most probably dealing with a non-sensitized patient and your immunological risk for rejection, pre-transplant risk category will be low risk, right? You come to the next category where you have T cell cross match as positive, maybe about 35% to 50%. The B cell cross match may be positive or negative. The HLA donor specific antibody MFI value is now positive and less than 5000. So it is anywhere between 1000 to 5000. And the Luminex single antigen bead may be indicated in this case, and you may find a donor specific antibody in that case. And the patient definitely will have a history of some sensitization in the form of transfusions, in the form of past pregnancies or abortions, or even a second transplant, maybe a previous transplant was done. So if you have such a result, then this patient will be categorized as intermediate risk. And then you come to a scenario where your MFI value is more than 5000, the CDC is strongly positive, and you have a strong single antigen bead test result also, and it is donor specific antibody. That means this will be a high risk patient. So this is how you use or you translate the laboratory results into clinical practice and clinical decision making, right? So for solid organ transplants, basically what we do is for establishing the relationship testing, which is a legal requirement, we do the HLA typing of the donor and the recipient and note down all the legal details and their relationship status and other things, etc. And we also do a CDC cross match, one before the transplant, and then when the patient and the donor is seen first, and then just before the transplant within 48 hours of the transplant surgery. That is the basic. But we also do a solid phase based or a Luminex based cross match or a flow cytometry based cross match. Then after the transplant, for monitoring if it is if there are some sensitizing events, etc., you may choose to do again one of these tests, and on a regular basis you follow up this patient with one of these antibody screening tests, right? So we've seen most of the other technologies that are commonly used. Let us look at the flow cytometry. Basically, again here what we are looking at is a reaction which is detected or which is quantitated using the flow cytometry principle. This reaction is between the recipient antibodies to the donor cells, right? So I would not go into the details, but here what we look at is the difference in the mean channel fluorescence between your negative control and the test sample, along with the other clinical and the laboratory criteria that you get as a part of the history and the patient case, right? Okay, so here we look at the MCS or the mean channel shift. That is the terminology that is used for understanding the results. Like there we had the MFI values, here we look at the mean channel shift values, right? So why basically flow cross match has evolved? That is because the CDC cross match is, though it is a universally acceptable tool and also a gold standard, but as I said, it has got very low sensitivity, right? Whereas flow cross match is more sensitive and also quantifiable method for any anti-donor antibody. If it is present, you can detect that, right? So this basically reduces the subjectivity and also inter-observer variability, because finally a CDC is reported with your own eyes. So there is a lot of subjectivity that is involved. You do a microscopic interpretation of cell death versus viability, whereas with flow, you will get a proper objective result. Okay, so when you do a flow cross match, again, it's a T cell reaction and a B cell reaction that is quantitated separately. And if you have negative T cell and negative B cell reaction, then there is no antibody and you are good to go for a transplant. If your T cell and B cell is positive, you definitely are dealing with a sensitized patient. There is a good chance that there will be a class 1 as well as a class 2 HLA antibody. If your T cell is negative but B cell is positive, then you are dealing with an isolated HLA class 2 antibody, or the patient has received some medication drug. So history is very important. And the last, if your T cell is positive but B cell is negative, then you may be dealing with low titer class 1 or a non-HLA antibody. So there are certain non-HLA antibodies like the MIC-A and the MIC-B that are most commonly tested for. So you can look for those also, right? So we've talked about solid organ transplant. There is also a lot of increase in the hematopoietic stem cell transplants these days. And for want of fully matched donors, now the haplo-identical transplants are on a rise, especially in the last 5 years. If you look at the data, a huge number of haplo transplants are happening and excellent results. Now, for this, the role of your proper donor selection is very, very important. So in the donor selection, apart from your routine donor-specific antibody screen and a proper, matching of the HLA types. So you do a high resolution typing of the donor and the recipient and a full proper match. Or if you don't have a full 100% match, then you do a full haplotype mismatched donor transplant, etc. Right? And you can also use certain other tests for your donor selection, like the KIR antigen testing. Right? So there are certain killer immunoglobulin-like receptors, and you test for them in your donor. So these are the various considerations that we make in order to choose a proper donor, because everything lies in proper appropriate donor selection when you are doing a haploidentical hematopoietic stem cell transplant. And then once you do that, once you screen your you screen your recipient for any donor specific antibodies, look at the MFI values. If the MFI values are negative, that means less than 1000 or so, nothing to worry. Or you do a risk stratification, and accordingly you may want to, just before the infusion of the stem cells, you may want to go for a therapeutic plasma exchange, a series of them, and reduce the donor specific antibodies and then infuse the cells, etc. So there's a lot of role of the transplant laboratory, the transfusion medicine, and a bone marrow transplant unit. Real true multi-speciality approach is what is required over here. So with that, let us now shift our attention to the other clinical applications, which will include disease association, platelet transfusion, trolley risk reduction, various adverse drug reactions. So these are various other scenarios where your HLA-based testing is indicated. So just to enlist them, for disease associations, there are various diseases which have strong associations of with various MHC class 1 genes, like for example, multiple sclerosis, leprosy, Rh isoimmunization, psoriasis, ankylosing spondylitis, right? We all know about HLA-B27, etc. Or with MHC class 2 genes, like again, multiple sclerosis, psoriasis, SLE, asthma, HIV-related diseases, thyroid carcinoma, Kawasaki's disease, celiac disease, etc. Right? When you deal with a refractory platelet transfusion, a patient who is refractory to any sorts of platelet transfusion, then what you do is there is a possibility that the HLA matched platelets, which is again, actually technically HLA matched platelets will be very practically impossible, but what you can do is screen the family members or give a trial of the platelet donated by one of the close family members. And because of the chance matching of one of the haplotypes with the recipient, you will have a better outcome, better survival of the transfused patient. So that is how you plan your platelet transfusions. You can even do applet matching for these kind of refractory patients. Similarly, trolley risk reduction again can be done with proper HLA matching in certain cases who are more prone to trolley. And there, along with the HLA, you also have to look at intricately at the human neutrophil antigen. So granulocyte immunobiology has now evolved in the last 3-4 years actually as one separate sub branch actually of our immunogenetics. Then as far as adverse drug reaction is concerned, I thought it is worth mentioning here about abacavir, which is used for retroviral therapy. And there you look at HLA-B5701, which plays a major role in, hypersensitivity reactions to abacavir. Carbamazepine therapy. So before you start that for epilepsy, you look at the presence of HLA-B1502, or allopurinol, which is used for gout or hyperuricemia. You look for presence of HLA-B5801. So these are certain clinical applications of HLA typing. Before I finish, just a practical aspect that for setting up a HLA laboratory, you will need at least a good space of about say 200 to 250 square feet, and you need to separate your areas, because pre-amplification and post-amplification should be separate, and there can't be any passing from post to pre, and contamination has to be avoided. A very good environmental surveillance is required, right? This is just a list of the tests that we offer at our HLA laboratory at KDAH. And apart from the service provided to the bone marrow transplant patients and the renal transplant patients, etc., there is also a specific role played for cadaveric transplant. So before I end, I just wanted to sensitize you about the deceased donor program, where basically the need of the hour today in our country is that many people, the public, should be aware that you can donate your organ and you can pledge your organ, and there is a process that is involved in pledging these organs. And organ transplant, the deceased donor program, has to pick up really, really well. There are some states, especially the southern states, are doing well in the deceased donor program, but as a nation we still have a lot of scope to improve. So what you need to know in this is the Transplantation of Human Organ and Tissue Act, the THOTA Act, which was defined in 1994 basically, and it legalized the concept of brain death, thereby facilitating the organ procurement. It aimed at stopping the commercial dealing of organs, all those mishaps that we were coming to know through newspapers, etc. And it defined the donor types like live related, live unrelated, and cadaver donor scenario. So the THOTA Act is very, very important. And also you need to know that we have a National Organ and Tissue Transplant Organization under the aegis of the NOTP, and that is known as the NOTO, and same you have in the state also. So there is a State Organ and Tissue Transplant Organization, so NOTO and SOTO. So I just want you to be well versed with these terminologies. There is a lots of legalities and guidelines and all that you can go through if you visit the websites of these. And the last concept is knowing something which is known as the cold ischemia time. So what this refers to is the amount of time that an organ is chilled or cold and not receiving any blood supply. Now this cold ischemia time, understanding that is very, very important, because beyond that time, if you are, or within that time, if you are not able to, reach the donated organ to the recipient, because your donor may be in hospital A, which is at one corner of the city, and the recipient may be, in one of the ICUs in the other corner of the city in another hospital, then you have to know the cold ischemia time, because after retrieval of the organ, this organ has to reach the recipient hospital at the earliest, and the topmost is heart, because heart has only a time available of only 4 hours, the cold ischemia time. For lungs, it is around 4 to 6 hours, right? So therefore you must have heard, in fact in movies also it has been now featured, that green corridor, how the green corridor was achieved, and how the heart was transported from one corner of the city, or from rather one neighboring city to the other city. And we did these kind of transplants, even cardiac transplants, for challenging, say, Rh negative patients also, even during the COVID pandemic, right? So a good understanding of the cold ischemia time helps us in streamlining these processes. And the role of the HLA laboratory over here is that even before the organ can arrive, the blood sample arrives of the donor, and you have the recipient from various hospitals. So the zonal transplant coordination team, they look into this, and there are different HLA laboratories that are identified and authorized for the testing of these samples. So we do basic testing like the CDC test, and also the donor specific antibody, or even a single antigen bead if the particular recipient is heavily sensitized, and things like that. Another noteworthy mention over here is that your HLA laboratory needs to be NABL accredited. That's kind of a legal mandate if you're dealing with HLA typing, especially in the live related transplant scenario. So that's it, friends. I think I've been able to cover the technical, a little bit of historical and practical applications and scientific aspects, as well as a little bit about organ donation. So thank you so much for this opportunity. And I would rather invite Dr. Naveen. I think all of us know him very well, and he deals with a lot of bone marrow transplants. So I'm sure he has got a lot of comments to make about his experience and the importance of the HLA based testing. So I would invite Dr. Naveen to share his thoughts on the histocompatibility and immunogenetics aspect. Thank you so much.
Thanks, Rajesh. Actually, I have no comments. You've covered it so beautifully and in such a simple language that I think hopefully most who are there in this webinar would understand. I mean, it's really very, very nicely done. The important thing to probably understand for either those who are in laboratory or those who are in clinics is that you have to be very careful when you interpret the HLA, and you have to be doubly sure before you release reports that that's exactly the HLA.
Absolutely.
One of the most difficult issues become when we don't pay enough attention before the release of the report. A laboratory report of creatinine of 1.5 and the next day it's 1.2 does not matter much. But one allele or one antigen you've written wrongly, and that's the end of it, for the simple reason there will either be a graft rejection if it is a solid organ transplant, and if it's a liquid organ, that means bone marrow transplant, it could be rejection or a horrendous graft versus host disease, and both would be equally ominous for the patient. So while the mismatch in the organ transplant is associated with rejection, the mismatch in BMT is either rejection or graft versus host disease, either of the two, and one cannot really predict which way the immunological vector will go. As far as bone marrow transplant is concerned, I think there is no longer the era of doing a 2-digit typing. It is always a high resolution, which is either by Sanger sequencing, when you are sequencing the exon 2 and 3, or you do the NGS based technique. So that NGS is more productive. You can take many samples together and do the typing. You need a very good knowledge of bioinformatics in it. Either you need to have someone or maybe acquire knowledge yourself. And that's the way to go forward for bone marrow transplant, to do the high resolution typing in all patients, because we see that though in sibling transplants, which are fully matched, one may do with intermediate typing, that's the 2-digit typing by SSP or the SSOP, which you have alluded to. But when there is homozygosity between in the patient or the donor, especially when there is consanguinity, we can get into trouble. So correct. The important thing to understand is the high resolution typing is very, very important in bone marrow transplant. As far as organ transplantation is concerned, I don't know much about it, but I believe the SSP or the SSOP should suffice.
Yeah. In
Not
fact, for organ transplants also now there is a move, especially for the renal transplants. There is a move to go beyond the AB and DR, because till now it was only the AB and DR testing that was done. But now even beyond that, because many patients develop antibodies beyond AB and DR. So there are a lot of DQ antibodies that have been reported now in patients who have transplanted. So even in the renal transplant scenario or the cardiac transplant scenario also, now the physicians or the surgeons, they require a complete ABC, DR, DQ, DP and at a higher resolution.
Yes. And the last comment what I would make is just like there is a registry for solid organ donations. So there are registries worldwide for the unrelated donor registries for bone marrow transplantation. And India itself has at least 5 unrelated registries which are functioning. One is in Mumbai itself, which is known as the Marrow Donor Registry India, which was started at Tata Hospital. And then there is one in Chennai, there's one in Bangalore, and there's one in Delhi. So there are already 4 unrelated registries functioning in India. And worldwide, there are more than 70 or 80 registries. And just like there is a cold ischemia time for organ transplant, there is not a cold ischemia time for stem cells for bone marrow transplant, but you can keep the stem cells in 4 to 6 degree or 4 to 8 degree for around 72 hours without losing the potency of the stem cells. After that, there is a loss of potency, and you need to cryopreserve the stem cells if you want to use it after a few months or years. I think that's all from my side. It's a very nice presentation, Rajesh.
Great.
Very difficult talk and very lucidly done. Thank you.
Thank you so much. So, do we have questions?
Yes, sir. We have one question in the chat.
Mm-hmm.
One participant has asked, could you elaborate on KIR ligand testing and how do we look at KIR mismatch in bone marrow transplants?
Well, I think Naveen will be able to answer much better, because they do, in fact, they were the pioneers in doing the KIR ligand testing at Actric, and there is a good publication also, I think, or I mean, we have looked at the results also. So, Naveen, would you like to take over?
So, the KIR ligand phenomenon sometimes I feel is overhyped, because most of the data across the world is still, the jury is still out whether it makes a major difference or not. And we have just recently analyzed our own data, where actually we have really not been able to see whether the outcomes had been different because of different KIR ligand mismatch. Now, what is KIR? KIR is the killer immunoglobulin receptor, which is present on the NK cells. And what is the KIR ligand? The KIR ligand is basically the HLA B or C, sometimes HLA A. So when you have an NK cell with a KIR receptor and you have HLA molecule on cells, predominantly the T cells and other cells, where you have HLA B or C, now you can group the HLA B and C into various ligands according to the amino acid sequence at a particular position. So those are known as epitope. So all the HLA-C can be clubbed into C1 or C2, and that's why it's known as C1 ligand or C2, all the HLA-C, and the B can be clubbed into BW4 or BW6. I mean, it's a bit, it's not complicated. It's just that, one has to understand the nomenclature. Now, when you have a KIR receptor, which finds its cognate ligand on a particular cell, and here you want to have that cell as malignant cell, the KIR is by default inhibitory. So if you have an NK cell with a KIR receptor which binds to the HLA on the cancer cell, and if they bind well, then there is inhibition, and the NK cell cannot kill the tumor cell. Now, if the KIR receptor does not find its cognate ligand on the cancer cell, then there is default activation because there's no inhibition, and that is and then the tumor cells get destroyed. So while it looks very logical, it looks very intellectual, I think in the real scenario, I'm not very sure how much difference it has really made. We do look at the KIR ligand and the KIR receptor, but we don't swear by it, at least in our center. I think more important is the KIR haplotype. So KIR is in the chromosome 19, and it also has 2 major haplotypes. One is known as AA, where you have many inhibitory receptors in the KIR, and you have B or BX, where you have many activating receptors in the KIR genotype of a particular donor. Now, if you have an activating donor type, that is BB or BX, then you probably have a good KIR repertoire in the donor, and that may help in decreasing the relapse. So just to cut a long story short, the haplotype of the donor, whether it's AA or BB, again, if you choose the donor according to this KIR genotype, you may do better if your donor is a BB or a BX haplotype rather than an AA. So I think that's the only thing where I feel that probably there's some effect of the KIR. Otherwise, all the models of KIR receptor and ligand, I am not very sure whether it probably makes a major difference, though you will find literature either way on both.
Correct.
Thank you,
I think very appropriately explained, and the clinical side, I mean, decision making basically. So we do have associations and correlations that have been reported, but finally a consensus has not really been made in the clinical significance of KIR ligand testing.
Thank you, sir. Sir, there is another question in the chat. Why are RBC tolerated without expression of MHC1 molecules?
Why are
RBC is tolerated without expression of MHC1 molecules.
so RBCs are tolerated in the sense we also have mechanisms where the antigen which are present on the RBCs, they trigger the production of various antibodies, like we saw, in one of our previous seminars, that you can have various antigens that are present on the red cells, which will lead to production of antibodies by the same immunological phenomenon, isn't it? Only thing is there is no rejection of the red cells that happens. But yes, the antigen is capable of mounting an immune response.
I think that is all, sir. That is the only questions we have in the chat box.
Great. So do we have Dr. Sumit or Dr. Muna?
Yes, sir. Thank you, Dr. Sawant and Dr. Khatri, sir. Just a small reminder about the upcoming talk on Thursday about investigating the transfusion-related problems, case-based approach by Dr. Preeti Desai. And I would like to share this QR code with you all. Please download the Telegram app and join the app, join our group through this link so that you get updates about the upcoming talks. Thank you all for attending.
Thank
Good
you.
night.
Thank you so
Thank you.
much. Thank you, organizers, as well as all the participants. Thank you so much. Thank